Hey there, all you cool cats and kittens. It's a Wednesday afternoon here at the Perrysburg Wastewater Treatment Plant, and I am about to get into some of the material in Chapter 7 in the OTGO workbook, which covers process control testing. So let's go ahead and get started. So I guess first of all, let's cover some of the reasons that we actually do testing here at the wastewater plant. Uh, Of course, first and foremost, we want to be able to measure how the plant is performing. So by testing raw influent coming into the plant and comparing it to our final effluent, that gives us an idea of how the plant is performing. Um, some of the tests that we perform, we have to report to the EPA. Um, all of our reporting to the EPA is self-reporting. Um, Of course, they're an agency that has to oversee hundreds of locations around the state, so they don't have the time or the resources to do any of that testing themselves. Um, Also, we might test to establish a historical database, which would allow us, when the plant is not operating properly, to maybe go back and compare current test results to past results to see if we can find a discrepancy and maybe help us figure out if we're having issues, maybe why those issues are occurring. Um, of course, comparing test results over time might help us recognize changing conditions uh, in the plant or maybe in the raw influent coming into the plant, maybe something going on that we might not recognize otherwise, that we might not be able to see uh, just through general observations. And lastly, at least here at my plant, some of the testing we do helps ensure that the plant is being monitored on a regular basis. Of course, it's a big plant with a lot of equipment, a lot of buildings, a lot of tanks, and a relatively small staff. So by requiring some of this testing to be done on a daily basis, we are ensuring that somebody is out in the plant looking at the equipment and at the tanks on a daily basis, and sometimes might notice an issue that otherwise would go unnoticed until that issue got larger. So those are some of the reasons why we might do testing. Of course, the chapter does talk about different types of sampling, grab samples as compared to composite samples. Of course, a grab sample is a sample taken from a specific location at a specific time. Those samples, of course, only offer a snapshot of what's happening exactly at that moment in that location. Um, some of the grab samples that we take here at the Perrysburg plant have to be taken for reporting purposes, and they have to be taken a minimum of 3 hours apart. Um, of course, when taking grab samples, in order to spot a trend, it's helpful to take the sample in the same location at the same time every day so that you can compare results. But I would also point out that it's also a good idea to occasionally take a sample either from a different location or at a different time. Sometimes if you take it in the same spot at the same time every day, that can be misleading if you're trying to spot something that's occurring over a 24-hour period. Composite sampling, of course, is exactly what it sounds like. We're taking several separate grab samples and putting them together to represent what is occurring over time, usually over a 24-hour period. For the purposes of reporting on our NPDES permit required testing, usually that time period is 24 hours, and usually those samples have to be flow paced, which means that when the flow is higher, more of a sample is collected. During lower periods of flow, say in the middle of the night, a smaller amount would be collected, so that when you put that composite sample together at the end of the day, it's going to be representative of what transpired over that 24-hour period. Process control testing versus NPDES reportable testing. There are some tests that we do that are strictly for process control. Other tests can be both. We do them and use them for process control, and they also have to be reported. The main distinction I wanted to make here is for the reportable testing, you have to use methods which are deemed acceptable by the EPA. All results of those reportable tests have to be reported. So say, for example, here at our plant, if we have to do a test 3 days a week, a minimum of 3 days a week. If we actually did it 5 days a week, we have to report all 5 of those results. We can't just say pick the 3 best ones. Also, EPA has divided each month into 4 different sampling weeks based on the day of the month. So it doesn't matter what day the month starts on. Our first sampling week would begin on the 1st and end on the 7th. Second week begins on the 8th and ends on the 14th, and so on. So here at our plant, if we have a test that needs to be done 3 days per week, we would have to collect 3 samples in each period. Finally, for these reportable tests, a couple things to point out. You've got to keep accurate records. Records need to be kept in ink. You have to record who collected the sample, the time and date that the sample was collected. If you make any mistakes on any of your lab data sheets or calibration logs or anything, they want you to draw a line through the mistake so they can still read what it said originally, and then write the correct information. You're not supposed to use whiteout or erasable ink or tear pages out of a notebook, anything like that. They want to be able to see if you did make a change, what change was made. And finally, equipment needs to be calibrated to the standard set forth by the manufacturer. So move on to just a list, an example of some of the tests that we do here at the plant that probably would be pretty common to most wastewater plants. We would do total suspended solids, mixed liquor suspended solids, carbonaceous biochemical oxygen demand, pH and dissolved oxygen, percent solids for sludge, also percent volatile solids, ammonia. Those are all tests that we do here in our own lab. Some of the other tests that we do that we would send out would be fats, oils, and greases, mercury, phosphorus, metals in our sludge. So we do use an outside lab to do some of the testing, and some of it we actually do here at the plant. Finally, my last 2 slides, I'm going to give you just a brief overview of how some of these tests are done, primarily so that when you do the math problems, you can kind of have it visual in your head. Let's start with the TSS, or total suspended solids. Here at our plant, we do this in samples collected in 3 different locations. Raw influent and final effluent we have to do for reporting purposes. The primary effluent we do just so we can get an idea of how effective our clarifiers are. We can calculate removal efficiencies based on that. So this test is done by passing a sample through what's called a Gooch crucible, which if you look at the picture in the middle is the little, real small dish here. There's a little paper filter in the bottom of that. The bottom of the crucible has holes for the water to pass through. And we would take a sample. We would weigh this crucible on the filter paper, and then we would pass a sample through it, filter out the solid material, and then weigh the crucible again after putting it in a drying oven to evaporate any water that might have collected in the filter. Depending on the concentration of the solid material, that's going to affect how much you can pass through the filter. I know, say, typically for our raw influent, we're lucky to get 25 to 50 milliliters through that filter. Primary effluent, usually we can get 100 milliliters through the filter. Our final effluent, anywhere between 250 and 500 milliliters through the filter. As the filter collects solids, it starts to blind off, and you don't want to put so much sample through it that you can't get all the water to pass through the filter. So this slide has a little example of how the math would be done. So when we weigh the crucible, the scale is going to weigh the filter in the crucible in grams, and we want our ultimate answer to be in milligrams per liter. So we have to convert the weight of the filter paper and the solids collected. We have to convert that from milligrams into grams into milligrams, excuse me, to get the number that goes at the top of our equation, milligrams. And then the second thing that we need to do is we need to convert the amount of the sample that we put through the filter, say in this case 500 milliliters. We need to convert that to liters because we want our final answer to be in milligrams per liter. So our first step would be to convert the weight in grams to milligrams. And then we need to convert milliliters to liters. And then finally, of course, we divide milligrams by liters to get our ultimate answer. So mixed liquor and return activated sludge suspended solids are pretty much the same concept. We use a bigger vessel called a Buchner funnel and a larger filter paper that we can actually pull out of the funnel after we pass a sample through it and put it in the drying oven to dry by itself. But again, the concept is the same. We need to convert the weight in grams to milligrams, and then we need to convert the amount of sample that we put through the filter into liters in order to do our final calculation in milligrams per liter. Sludge percent solids and percent volatile solids. This test is done with a dish. We would put a sample of, let's say, raw sludge, which would be this dish on the right, in a dish. Our initial step would be to put it in the drying oven to let the water evaporate. We would, of course, weigh the dried sludge. We would weigh the wet sludge sample, and by dividing the dry sludge by the initial weight of the wet sludge, we would be able to figure out what percent of that is solid material. The second step in this test is to convert this into percent volatile solids. We want to know what percentage of those solids are actually volatile content, which we use volatile content to determine how much of it is organic. So to determine volatile content, we would actually take our dried sludge sample and put it in a furnace and burn it. at 500 degrees. And what's burned off, which I'll circle in red here, that's what's left. That's called fixed material. You can think of it as inorganic. And then by dividing our fixed material by the dried sludge that we started with, that will tell us what percentage of that dried sludge is actually uh, fixed material or inorganic. So biochemical oxygen demand, this is a test that we use to kind of estimate how much food is available for the microorganisms in our wastewater. We've touched on this test before um, and did the math. Basically, what we're doing is putting uh, our sample in a 300 milliliter bottle. That's the standard bottle size. This bottle here on my slide uh, was for a raw wastewater sample. And looking at the bottle, it looks like Terry put, excuse me, 25 milliliters of sample in that bottle. So uh, in setting the test up, we would have, I guess, about 275 milliliters of water that she would prepare to put in the bottle, and then 25 milliliters of actual sample. So we fill the bottle all the way up to the top, and then we're going to measure the dissolved oxygen in that bottle at the start of the test. Then the bottles go in this incubator over here for 5 days. And then we're going to pull them out of the incubator and we're going to measure the dissolved oxygen level in the bottle again. And then whatever oxygen is used up is what we're going to plug into our formula to calculate biochemical oxygen demand. Two things that you might need to know for the exam would be 5 days. The incubation period is 5 days. And then the second thing you probably are going to want to know is that the temperature of the incubator is 20 degrees Celsius. And that is a question that is pretty common on these exams. What is the BOD incubation temperature? 20 degrees Celsius. So just a few more finer points of the test. When she sets these up, there are always 2 blank bottles that don't have any, should not have any BOD in them because they're just basically distilled water. So we should not see any oxygen depletion in those bottles. We also test a known standard, which should have a predetermined amount of food in them for the microorganisms. So after 5 days, that oxygen depletion should be within a certain range, a known range. And so what we're doing when we test our blanks in our standards is basically what that's telling her is that she set the test up right and we didn't contaminate any bottles or anything like that. When we do the testing on the standards and on our final effluent, we actually need to seed those bottles with microorganisms, because if you think about it, there should not be any microorganisms in our final effluent after it's gone through disinfection. We also seed the standard bottles so that we can make sure that we put some microorganisms in the bottle to actually consume that organic material and use up oxygen in the process. So this test is basically we're measuring how much oxygen is being used up while the microorganisms are consuming that organic material. So the more organic material that is in the sample, the more oxygen is going to get used up by the microorganisms. So that's a little concept of the test. Dissolved oxygen and pH, these would be grab samples. These are two things that we need to report to the EPA 5 days a week while the plant is staffed. In our case, we have to do it a minimum of 3 hours apart, 3 samples a day. If your plant is staffed 24 hours a day, then you probably need to do it once per shift. So for this test, we have a pH and dissolved oxygen meter. Actually, it's 2 different meters. All the software is contained in the parts that get suspended in the flow, and then this part here just stores the information and displays it for us. So in this display model, we're able to store the test results. We're able to store who actually did the test and where the test was taken, what time it was taken. So that helps us maintain our records for EPA should they ever want to look at them. So